The eLife Podcast from eLife, the open access journal for outstanding research in the life and biomedical sciences. Online at elife.elifesciences.org.
Hello, welcome to episode 25 of the eLife Podcast produced by The Naked Scientists. I'm Chris Smith. This month, how carbon causes lung disease, why sleeping benefits your memory, and the mathematical model that adds up to antibiotic resistance. First, though, to the deep oceans, where Gillian Peterson from the University of Vienna has been looking at how mussels team up with microbes that feed them. Genetically, these bacteria are endowed with a hefty arsenal of toxins, which they might use to defend their mussel hosts, or just to start a chemical conversation with them.
These mussels are found all around the world in the deep sea at oases of life at hydrothermal vents. They're the most dominant animals that are found at hydrothermal vents. And they're able to live there because they associate very intimately with a certain type of bacteria. Now, the deep sea is known to be very nutrient poor. There is very little food in the deep sea for animals. But it's not true that the whole deep sea is completely devoid of food. In fact, you may have heard of hydrothermal vents. You may have even seen the images of these impressive black smokers, fluids that look like black smoke pouring out of a chimney. In fact, it's pouring out of the seafloor. This black smoke is composed of a toxic chemical mix of things like hydrogen sulfide and methane. And this is actually an excellent food source for bacteria. And the only way that animals can live in these extreme environments is by teaming up with bacteria that use these chemicals to power primary production. And through this chemosynthetic primary production, they can feed their hosts.
Where do the bacteria live in the host? Do the bacteria live outside the host and the host eat the bacteria? Or is the relationship more intimate?
This is an extremely intimate symbiosis. The bacteria live inside of the cells of the animal, and they're hosted in an organ called the gills. And this is usually the organ that marine animals use to breathe underwater. But in this case, the organ has become much larger because it's housing an enormous population of symbiotic bacteria.
And these bacteria, are they only found at these hydrothermal vents, or do they have a lot of factors in common with bugs that we would find elsewhere all over Earth?
We were trying to understand more about these symbiotic bacteria, so we did some genome sequencing. And when we compared the deep sea symbiotic bacteria to all other known bacteria, let's say pathogens or environmental bacteria, we were surprised to find that they very much resembled pathogenic bacteria that are known to cause disease in humans, for example. They encode an enormous number of toxin-like genes in their genomes. And this was unexpected because we know that these bacteria are beneficial for their hosts.
Where do they come from then, these microorganisms? Did they start life as pathogens and have found their way into the deep sea? Or do you think the equation worked in the opposite direction?
For the longest time, researchers thought that symbioses may have evolved from pathogenic interactions, that first bacteria became pathogens, and over time they learned to live in cooperation with their hosts and eventually might have become beneficial. But our study and many others of symbiotic bacteria have shown that these beneficial bacteria actually far outnumber the pathogenic bacteria on our planet and even on and in our own bodies. So it may be that pathogenesis is the exception, and these bacteria first evolved to help us. And in very rare cases, they actually harm us.
How do the toxins that you found in these symbionts work? What are they doing in the context of the mussels that they associate with?
The way that we look at this is to compare them to genes that are, or proteins that are very similar, where the toxic activity has actually been shown in laboratory studies. Many similar toxins have been shown to mediate communication between animals and bacteria. Ours may have a similar function. We know that the bacteria need to be recognized by the host. There are millions of species of bacteria in their environments, and yet they can recognize their own symbiont out of this myriad of environmental bacteria. And then the bacteria need to invade the host cells. So there needs to be mechanisms of recognition and communication where a symbiont says, "Hi, here I am," and a host says, "I recognize you, you're a friend, please come in." Another role that these toxins might have is to protect the symbiosis, so to protect the host from its natural enemies. And this is something that has been overlooked. There have only been a couple of studies investigating pathogenesis or parasites in the deep sea. These deep sea mussel beds can be up to 5 times as dense as the mussel beds you would find in coastal regions. And this is the perfect oasis for a parasitic organism. And there's very little known about how they manage to defend themselves. And that's our second hypothesis, that some of these toxins might be involved in defending the host against parasites. And this came up because we noticed one particular species of mussel that has, over evolution, lost the symbiont that encodes all of the toxins. And this particular mussel species is very heavily infected by a parasite, a trematode parasite, that infects the gonads and eats all of the reproductive tissue of the mussel. It's a pretty nasty parasite. So this is what we call a defensive symbiosis, that the symbiont may be able to defend the host against a common enemy.
Gillian Peterson. Thanks to decades of effort, Richard Doll and his colleagues proved that smoking is bad for your chest. In fact, the lungs of very heavy smokers become stained black or brown with carbon deposits. But no one knew whether this was a cause or just a consequence of various other smoking-related conditions like emphysema or chronic obstructive pulmonary disease, where sufferers find it progressively more difficult to breathe. Now, Farah Karadmand at Baylor College in Texas has discovered what that brown pigment is. By exposing mice to a pure form of it, she can trigger lung disease identical in many ways to that suffered by smokers and coal miners. So it looks like a major culprit in chest disease is just carbon.
The story began when a graduate student in my lab isolates a particular cell type from the lungs of humans who've smoked. That particular cell just appears to be dark in color. And repeatedly, he came to my office and showed me these cells. And repeatedly, I very much explained that this is what's been explained in pathology textbooks, that smokers collect dark material within their lung. And so his next question for me was the origin of this dark material. And I referred him to pathology textbooks that describe this as anthracotic particles, the nature of which was rather elusive. And so he went on to disintegrate the cells, the result of which ended up being a very fine material that we really was visible by naked eye. That's when we contacted our colleagues over at Rice University. They decided to give the composition a try to understand what the chemical nature of this dark material was.
So what was that dark material? When you look in those cells, the stuff that's staining them black, what is it?
When I got a phone call back from our colleagues at Rice University, their first question was, where did you get these fine particles of 15 nanometer, which is 15 billionth of a meter, very small particles that are pure elemental carbon black?
So how did you then tie the presence of these particles in the lung to the disease process? Because they could just be there as an innocent bystander. The fact that they're in the smoke that the person breathes in doesn't mean necessarily they're connected to the disease process, does it?
The connection with the disease process was the particular cell types that these nanocarbon black collected. In fact, if you take the same cells isolated from the lungs of mice that have been exposed to cigarette smoke and transfer these cells to naive mice, meaning mice that have never seen cigarette smoke in their lives, a similar pathology starts happening in their lungs, meaning that there's quite a bit of inflammation and changes that are consistent with a smoker's lung is now shown in the mice that had never seen cigarettes. Therefore, for the first time, it connected a particular cell to disease formation.
How do you think that those literally just little particles of carbon are causing the disease then? What are they doing to the cells that they end up in that leads to that disease, both perpetuation in one animal, but also the ability to transmit that disease to another?
When nano-elemental carbon black accumulate in these cells become activated to try to undo the damage, meaning that they are now encountering a danger signal that they're trying to alleviate. And therefore, they turn on a whole program of genes that we have identified, some of which are important in DNA repair damage, for instance. When they turn these genes on, the result of which results in an activated cell that then can direct the cells of your immune cells that are normally there to protect you against foreign invasion or dangers, it can result in inflammation in the lung.
But how do you know that it's the carbon particles that are doing that per se, and not, for instance, something stuck to the carbon particles? Because when these come into the body, they're going to be laden with a whole range of other hydrocarbons and other materials from the burning cigarette or whatever other forms of pollution you've been encountering, aren't they?
This is an experimental model where we had access to highly purified elemental carbon black that was devoid of any material that could have possibly been attached to it. And they're all experimentals that were performed in my laboratory used particularly a form of elemental carbon black that is 99.999% pure.
It seems very surprising that given the ubiquity of fine particles of carbon in the environment, that the lung hasn't evolved a way not to respond like this, to clean itself out. Why do you think that's the case?
Well, in fact, the lung does clean itself out for the most part. And this is perhaps the reason why we find these heavy, dark stain material only in the lungs of heavy smokers. It's a matter of the amount of material that the lung can handle.
So it's once the system gets overwhelmed and can't remove the material sufficiently fast that the damage then begins to set in. That was Farah Karadmand. The UK's Chief Medical Officer, Dame Sally Davies, has ranked the threat posed by antibiotic resistance as on par with terrorism. And with good reason, because increasingly doctors, both in hospital and in the community, are having to switch to more intense and usually more costly antimicrobial drug regimes because formerly effective, cheaper drugs now often don't work. A number of strategies have been put forward to combat the problem, but one area being debated is whether to hit bugs really hard in one fell swoop or take a more gentle approach. Now, a mathematical model developed by Imperial College scientist Carolyn Collin suggests that both strategies actually have their places, but you need to use them under the right circumstances, as she explained to Greer Jackson.
So it's long been thought that a hard and fast approach to treating with strong antibiotics will be best at preventing the emergence of drug resistance within individuals. And there's also a opposing or competing view to that of course, when you treat very strongly, you also very efficiently select for any resistant organisms that might be present initially or that might arise during that treatment. So there have been two recent bodies of literature, one arguing for a hit hard and fast, treat really strongly with a strong dose of antibiotics, and the other arguing for a more moderate approach to using the least possible amount of antibiotics that will cure the infection.
And your paper is looking at these two strategies and seeing which is best within a mathematical model. How did you go about looking at that?
So we used two models. We used a model for the bacterial infection inside one individual, and we studied how two strains, a resistant strain and a sensitive strain, might interact and how that might play out under different strengths of treatment. And then we did the same thing at a community level where we imagine a drug-sensitive strain and a drug-resistant strain circulating amongst individuals, and we look at whether really strong treatment or more moderate treatment, which of those will drive more increases in resistance.
What did your model show?
So we found that both of these two opposing views can really be right under very sensible assumptions in models. So it can be the case that it's really best to do the hard and fast treatment, but it can equally well be the case that it's really best, in terms of minimizing resistance, to take a more moderate approach to how much treatment to give. And we found that the key factor that determines which of those is right is how effectively the two strains are actually competing with each other.
Competing? What do you mean?
So two strains would be competing if suppressing one of them paves the way for or provides additional resources for or in some way helps the other one. If the strains are just spreading independently and they're not related to each other in terms of how many you have of one and how many you have of the other, then that's a very different scenario in terms of whether treatment drives resistance than if the two strains are depending on the same resources and are really in strong active competition with each other.
So competition is the key thing here.
That's right. We found that when the resistant strain was a fit and strong competitor to the sensitive strain, in order to minimize resistance, it's best to do a more moderate approach to treatment. Whereas when that resistant strain can be assumed not to be a very fit competitor, either because it just doesn't grow very well or because it faces a really strong immune system that's really effective at suppressing it, then the more aggressive approach is great because it more rapidly suppresses the sensitive bugs and they're most of the problem and they don't have a fit competitor.
This is all in a mathematical model, so I wonder what the limitations of this might be. Is this something that you have seen within the real world in vivo?
Well, I think we have certainly seen rising resistance at population levels. So you can track the emergence of resistance with a short lag behind the introduction of new antibiotics. So we know that using antibiotics is driving resistance on long terms at the population level. And those are regimes where it's actually very hard to just empirically experiment. So I think there's a real role for mathematical models to play here in uncovering fundamental mechanisms and looking at what's important in driving these effects in a way that can maybe help us to guide empirical studies to understand these interactions.
I suppose in an ideal world, you would be able to test this sort of competition within individuals to determine what sort of treatment might be best. But I suppose that's not something that's already on the cards just yet.
Right. I think it would be really important to get much better understanding of the kinds of strain diversities that we have in human infections in lots of different pathogens. And also not just a picture of that diversity as a single snapshot, but to really develop an understanding of how strains are interacting within hosts and also in communities, of individuals. And I think there are some real challenges to that, but I think there's a growing awareness that resistant strains are increasing in fitness, and we need to start thinking about these possible competitive interactions between resistant and sensitive strains.
Caroline Collin from Imperial College. If you want to learn something really well, the advice has always been to sleep on it. But, according to Kinga Iglui, it looks like you also need to really value what you want to learn to get the most out of the effect.
We knew sleep had an effect on memory consolidation, but we did not know how reward and sleep interact, whether there is a selective effect of reward. We might have thought of it because we generally remember rewarding things better, like, for example, a great party that you had when you were a child. But it had never been tested how this works in the brain.
I suppose the evolutionary purpose of this is to prioritise the retention of information that in some way is likely to benefit an individual in the future.
Yes, it is. In evolution, we will try to remember things that are important, that we will need for our survival or for a successful life. So that is why we thought that rewards may help memory consolidation.
Tell us about the study design. What did you actually do in order to probe that?
So we make people come in early afternoon. They learn pairs of pictures preceded by a dollar coin or a cent coin. And we tell them that the dollar coin items will yield higher reward at the end of the study, so that they will be paid more if they remember the highly rewarded items better.
And where does the sleep
Yes.
come into it?
So after they have learned it, half of the participants take a nap and the other half stays awake. During sleep, we monitor their sleep using EEG, which is electroencephalography.
And then, having established that the person slept and what their brain activity is when they sleep or when they remain awake, you then present them with those cards again and see how good they are at remembering the pairings you asked them to learn earlier?
Yes, we show them the same pairs, and so we test their memory.
Right, well, let's look at the sleep first. Did the sleep improve retention of those memories?
Yes, it did. We had an overall effect of sleep, so sleep improved retention.
And then if you look at the value attached, was there a difference there too?
There is a difference between subjects who slept and subjects who stayed awake straight after, and there is also a difference between highly rewarded items and lowly rewarded items. Subjects remember highly rewarded items better.
And even better again if they sleep?
Yes. Then here we have to be clear at the test point, so straight after sleep, we do not have an interaction effect. So at test, there is only a straight effect of sleep. So subjects who slept remember items better, and highly rewarded items are remembered better than lowly rewarded items.
Is this just a short-term effect? Is this something which is manifest only on that day that you test them in the laboratory? Or if you pull people back days, weeks, months later, do you still see a preserved benefit through having had that sleep plus the high reward versus low reward situation?
Yes, it is important to stress that. We made people come back 3 months later, so a long time later, and we retested them on their memories, and there was still an important effect of reward, and selectively for the group who slept.
Putting this together then, how do you think this is working? And what did you see when you look at the brain activity? Does that give you any clues as to what the sleep is doing to make this effect manifest?
Yes, what we find is that there is an effect of sleep and of reward, so that you remember highly rewarded items better. And we have linked that in the brain to an activation of the hippocampus. So we find a higher activation in the hippocampus for highly rewarded items compared to lowly rewarded items for the subjects who slept versus compared to the subjects who stayed awake.
So if you want to learn something really well, you need to convince yourself that knowing it is extremely valuable to you and then sleep on it. That was Kinga Iglui from the University of Geneva. Sticking with the brain, can information sneak into your mind unconsciously and influence your decisions without you being aware of it? Previous studies had speculated that there might be two different thresholds. Some stimuli we are immediately consciously aware of, others might be biasing our thinking without us knowing why. It turns out, though, that this was just wishful thinking. Megan Peters.
For researchers studying consciousness and conscious perception, we want to be able to separate how well you can do on a task from your ability to consciously see the things that the task is asking you to do. The problem with this is that many times when you make something on a computer screen so dim that you can't actually consciously see it, you also remove the ability to completely do the task at all. And so it takes a careful experimental design to create a stimulus that you cannot see, but that you might still be able to do some task with. Because then we can separate your ability to do some task from the conscious experience of that task. And that allows us to do things like neuroimaging studies or brain scans, where you can say, okay, well, we held task performance the same. You can do the task just as well, but you say you're not conscious of it. So then we go and look for what makes something conscious or not.
There are therefore different thresholds. There's a threshold at which you are consciously aware of something, and a threshold at which your brain nonetheless knows it's there.
That's the theory, but what we're showing here with this study is that those two thresholds are actually one and the same. In this type of context, there isn't a difference in the threshold between when your brain becomes aware of something and when you consciously become aware of something. And this is very surprising and flies in the face of a large amount of research that's been done up until this point.
Very, very tricky to unpick that. How did you manage to prize apart those two and prove they're effectively one and the same?
The previous way of doing this type of research was to show someone a stimulus on a computer screen or have them listen to a sound stimulus and have them do some task and then report, did you see it or not? Or how sure are you that the answer that you just gave was correct? But there's a really big problem with this, which is that just because I tell you that I don't see the thing on the screen doesn't mean I actually didn't see it at all. Maybe it was just slightly dimmer than whatever I count as a threshold for reporting that I saw it. We wanted to get around this threshold problem by designing a task where you do 2 judgments to 2 different stimuli, and then you indicate which of the 2 judgments you are more confident about. So this way it doesn't matter whether I am very confident overall or very not confident overall. I still have to choose one to say I'm more confident about this decision than the other.
Because one acts as its own control in that way, doesn't it? You're comparing one against the other, so they are literally cancelling the noise in both, which is very elegant.
That's the goal. And the slightly clever thing that we did with this type of task is we had participants, again, make 2 judgments about 2 different stimuli, but one of the stimuli wasn't actually even there. So we showed them this stimulus on the screen. It's a gray and white stripy patch, and it's very brief, and it's masked by other colorful images that flash before and after it. And the task is to say, are the stripes tilted to the left or the right of vertical? And then we show you another patch, and we ask you to do the same task, left or right of vertical, but this time we turn the brightness, the contrast, down all the way to 0. So there's actually nothing there on the screen. And then we say, which of these 2 decisions are you more confident in? And if you can do the task where you can report left or right better than you would if you were just totally guessing, but then you can't tell us which one you're more confident in, then you really can't tell the difference between being able to do the task and being able to just guess randomly at nothing there on the screen. And so in this way, you have no introspective access to the information that contributed to your decision about that stimulus. And so in this way, we would say, okay, if that occurs, that is unconscious perception. And it turns out it doesn't exist at all.
Megan Peters, she's at the University of California, Los Angeles. That's it for this month. You've been listening to the eLife podcast, previous editions of which you can find for free on the eLife website. That's at elife.elifesciences.org, or you can subscribe on iTunes. This is a Naked Scientists production. I'm Chris Smith, and you can find out more about what we do on our website at nakedscientists.com. I shall be back next month with another look inside eLife. Until then, though, thank you very much for listening, and goodbye.
The eLife podcast from eLife, the open access journal for outstanding research in the life and biomedical sciences. Online at elife.elifesciences.org.